I have thrown a lot of information at you, a lot of words, difficult terms. Might have become hard for you to process all the information. The beauty of cell biology is you can see the things you are working with. So let me show you an animation, and hopefully a lot of these concepts that we have talked about will become very clear. So let's watch this animation. So here we have G-actin. G-actin is globular actin, or the actin monomer. On your screen, you can see it is in white. So G-actin, this is the ribbon structure of this molecule. It's a globular protein. This is the ATP molecule. It looks familiar. So ATP binds the G-actin. Now it is ATP actin. Remember, it is in yellow color. So once this molecule is, the G-actin hydrolyzes ATP, now it is in blue color. So ADP actin is blue and ATP actin is yellow. So these are monomers. They have ATP bound to them. So we are going to look at spontaneous nucleation, these monomers bumping into each other. There is the nucleus we've talked about. Once the nucleus is formed, the elongation is rather rapid. Now please note that as the polymer is polymerized, as the monomers are forming the polymer, rate of GTP hydrolysis is also catching up. So you can see part of the filament is becoming blue. So part of this actin is blue, which is bound to GDP actin, and part of this filament is the monomers are still in ATP form. So barbed end, or the plus end, is yellow. The pointed end, or the minus end, is blue. So filament, the monomers will keep on adding, and you will have a filament. So now you are going to watch treadmilling, which we talked about. What I had said, that the critical concentration for the ATP or ADP actin, so the blue is ADP actin, the critical concentration here is greater. So if there was a lot of monomer present in our media, or in our test tube, then the monomer will add at the blue side. If not, it will start to lose. So if you are below the critical concentration, you will lose the monomer. The filament will depolymerize. If you are at critical concentration, it becomes the rate of on is same as rate of off. So you are basically in a sort of a equilibrium. So if you are below critical concentration, the filament will become shorter. So blue part will get shorter, but the critical concentration for the T part of the filament it's still higher than the critical concentration. So yellow subunits will keep on adding at the plus end of this filament, and the blue part of the filament will keep on losing its monomer. Since the two rates are equal, it will appear that filament is moving. Actually, the filament is not moving. As you will see, it is basically exchange of monomers. So the blue part is getting shorter, whereas the yellow part is getting longer, and the net result is basically that you see that filament seems to be moving, although it is not moving. Ultimately, if there no net addition of monomer onto the polymer, the whole structure, your whole filament, will become GDP actin, and ultimately it can also depolymerize, and you will have no actin there. So the whole structure basically depolymerizes, and we have lost the polymer form. Okay, I hope this animation was useful to you, and you are now able to understand some of the things that we have talked about.